All right, good afternoon. My name is John Connolly. I'm one of the co-course directors for the MSc Regenerative Medicine. And thank you for those who are joining us for this afternoon's webinar. I'm just going to give people another minute or so to log in, and then we will get started. Okay, it looks like we have most people logged in. And so for those who are joining later, they can pick up wherever we are in the presentation. So as I mentioned, my name is John Connolly, and along with Dr. Kristen Brown, we lead the MSc in Regenerative Medicine here at Queen Mary. And so this afternoon, what I'd like to do for you is kind of give you an update on the program and the course, and also to highlight a few recent research developments and things of interest that are going on here at Queen Mary, and hopefully get you interested in this program and the university as a whole. Just a couple reminders about the presentation this afternoon. So we are recording it just so that you're aware, but this will be made available if you'd like to go back and look at it later on. Also, one other thing, so I'll be running through a short presentation. It'll last maybe 20 minutes, and then we can have a question and answer session at the end. So if you want to save your questions for the end, and then you can put them into the Q&A dialogue, and we can go through those at the end of the presentation. All right, I will go ahead and get started. So as many of you know, and you probably are well familiar with what's on our website, but just to remind everyone, the Master's in Regenerative Medicine is a full-time one-year program, and the focus is on the science and application of regenerative technologies, and basically how we can use stem cells, biomaterials, and a number of other different technologies to promote healthy tissue repair and regeneration. And because this is an interdisciplinary subject, it's delivered by two different schools, primarily by the School of Medicine and Dentistry, where we're based, but also with our colleagues in the School of Engineering and Materials Science. The program consists of 8 taught modules totaling 120 credits, and a dissertation research project totaling 60 credits. The structure is broken up into 3 semesters, each about approximately 12 weeks long. So the first semester runs from September through December, and in the autumn semester, there are 4 modules of 15 credits each. And these include topics such as the cellular and molecular basis of regeneration, stem cell and developmental biology, research skills and methodology, and advanced tissue engineering and regenerative medicine. Then in the second semester, students have a bit more of a choice. So we offer 5 different modules this upcoming year, and the students can choose 4 of those. And these include modules on ethics and regulatory affairs, biomaterials in regenerative medicine, which I lead, tissue-specific stem cells, induced pluripotent stem cells and genome engineering, and neurodegenerative diseases. And then finally, in the third semester, which runs approximately May through July, the students carry out an intensive full-time research project where they're working in the labs for approximately 12 weeks on a defined piece of work. And this can be in a lab based either in the School of Medicine and Dentistry, SMD, or the School of Engineering and Materials Science, SEMS. And the way that we work these projects is that early in the autumn, we distribute a list of the topics, and the students can read through those, set up meetings with the supervisors, and discuss the project a bit more. And then at the end of semester 1, the students submit their top 5 project choices. And then what we do is to try to match students up with one of their preferred topics. And these projects range from a wide range of subjects. They can be things such as the basic or fundamental biology of stem cells and developmental biology. It could be studying mechanisms of tissue degeneration or repair. And it can also be more applied. So for example, projects in the School of Engineering often focus on the development of biomaterials for tissue engineering applications. And so after the students select their projects, these are assigned early in term 2 so that they've got some time to do some background reading and get familiar with the subject. And then they start work in the labs, usually around the first week of May. And by this time, they'll be completed their, or they will have completed their second semester taught modules so that they can focus entirely on the research over the summer. And then this final project is assessed by a written dissertation, which is usually due in August. And so then upon completion of the master's degree, students graduating from this program go on and do a number of different things. And so probably the main route that students take is to go on and do a PhD. And students from our course have gone on to PhDs at Queen Mary, but also UK universities such as Manchester and Southampton, but also in international programs in places like the Netherlands and in Ireland. A number of our students also find employment working as research assistants, either in academia or clinical labs. Several have gone on to industry within the pharmaceutical sector. We've also have a number that have gone on to science publishing, so taking their scientific skills and using those in science publishing and communication. We've had a couple who have gone on to secondary school teaching, and a number that have gone on to further their education and train as clinicians in medical school. So there's a wide range of topics, but whatever you're interested in doing beyond the master's degree, we've got several mechanisms for supporting that, and that includes our LinkedIn alumni network, but also the Queen Mary Careers Office, which has a lot of resources for job searching and network building. So those are some of the kind of basics of the program and what our previous students have gone on to do. I would like to just quickly touch upon probably a question that's on everyone's mind, which is how will the COVID-19 outbreak affect the program in this upcoming year? So at the moment, our plan is to offer what's called blended learning. And so this will be where the modules in the first semester are delivered, the core content is delivered as online recorded lectures that the students can access and review in their own time. And then that's followed up by small group tutorials where we kind of consolidate some of those concepts, discuss things, and that's a live synchronous activity. And where possible, we'll be doing these small group tutorials in person and on campus. And of course, this will depend on distancing controls and maintaining safety where possible. And then we're hoping that on-site delivery will resume relatively soon, but that will be, of course, depend on the situation and how things progress. But we are hoping that potentially in the second semester, we would resume kind of normal on-site delivery. I think one thing to bear in mind, and everyone's probably well aware of this, is that the outbreak and the situation changes very rapidly, and it's also very difficult to predict. So we will be doing our best to keep people updated and adapt and change as needed. I'd like to just kind of point out one link here, which is on the Queen Mary website, and it's some guidance for students and applicants about the current situation and what will be available. Last point about this is that, regardless of what happens, whether we have to offer more online and less in person, or we can open up further, in either instance, we will support your education as best as possible and ensure that you still get the same education and knowledge. I'm sure you have questions about that, but what I'd like to do now is kind of go on and talk about some of the more current research, and we can come back to this point if you do have questions at the end of the presentation. Okay, so the next part of the presentation, I wanted to highlight a few different things that are going on within the university that are fairly exciting and hopefully of interest to you. The first is work that's been going on around the development of next generation engineered tissues. And this is primarily through the launch of a new center for in vitro predictive models. So basically using our tissue engineering tools and technologies to build in vitro models of tissues and diseases. And so I'll talk a little bit more about this in a minute. And then I'd also like to highlight a few areas where the university is contributing to COVID-19 research, particularly in the areas of screening, diagnostic technologies, and understanding the risk factors of the disease. So when we talk about modeling of human tissues and diseases, or how we study human tissues and diseases, there are kind of two main modes of research. One is in vitro cell culture, where you grow cells, typically on a plastic dish, and they're submerged in this rich nutrient medium, or the use of model organisms, such as the mouse. And while these have been, instrumental in, almost all of our biomedical breakthroughs over the past 30, 40 years, there are limitations. So for example, the in vitro culture is very simplistic and doesn't often capture some of the more important or complex interactions that we see within the body. On the other hand, animal models such as the mouse, they're more complex systems. They've got immune systems and multiple different cell types that communicate with each other. But at the same time, this is a mouse, and its physiology is much different than that of a human. And so as a result, these models are sometimes poor predictors of what's going to happen in the human body. And moreover, they're sometimes ineffective tools for discovery and development of new therapies. And this has been a major limitation for developing new therapies, and often why drugs or other biologics fail in clinical trials. And so because of this limitation, there's been a really big push in the last few years to develop more advanced in vitro models that better replicate what goes on in the human body. And so just to name a few of these types of technologies, there's what we refer to as organ on a chip, where you've got microscale fluidic channels with cells in them, and these can be very precisely engineered and controlled. We also have a number of technologies referred to as organoids, where you start with a stem or progenitor cell and allow it to develop into an organ of choice. And so, for example, this image here is a cerebral organoid that mimics brain development. And then there's also 3D bioprinting, where we use 3D printing technology combined with some of our tissue engineering approaches to build living cellular structures, such as the heart seen here, with very precise physiologic architectures. And so with all these types of technologies, some of the advantages of them is that we can mimic some of the more complex 3D structures that we see in the body. We can also incorporate multiple cell types, and so we can understand how they communicate with each other and how that changes dynamically over space and time. And then one of the most important aspects of these types of technologies is that they're human-based. We can use human cells and study how they interact, and hopefully they better replicate what goes on in the body. And like many places, Queen Mary is very active in this area of research. And recently we set up this new Center for Predictive In Vitro Models to kind of bring together all the different people within the university who are working on developing these technologies, but also applying them to understanding diseases and tissues. And so this is a virtual center, so it's not, doesn't have a physical building, but it brings together researchers from across multiple different disciplines and campuses, so both medicine, biological sciences, and engineering. And so if you are interested in some of this work, there's a website where you can find out a bit more about that. But I'd also like to highlight a couple of the core facilities associated with this Center for In Vitro Predictive Models. One is the CREATE Biofabrication Lab. So this is actually a facility that I started and currently run here in the Blizzard Institute. And this is focused on the use of 3D bioprinting to create more advanced tissue models and tissue replacements. And then the other key facility is the Emulate Lab. And so this is focused more on the organ on a chip technology. And so this is a collaboration with the company Emulate, which is one of the founders of the organ on a chip technology, and uses their systems. And this is based down in the engineering labs at the Mile End campus. And so these two facilities together are really trying to drive forward new developments in tissue modeling and disease modeling. And so to kind of celebrate the launch of this new center, we're hosting an afternoon symposium. So if you are interested in some of this research and science, it's going to be held on the 22nd of July in the afternoon. And it will include a series of talks, one from a keynote speaker, Professor Jennifer Lewis from Harvard University. There'll be several talks from group leaders and leaders in the field here at Queen Mary. And then there'll be a series of kind of short, rapid fire talks from students and postdocs, more junior researchers involved in this type of work here at Queen Mary. So if you are interested in this, I'd encourage you to attend. It's free, and you can register through this Eventbrite website. So now moving on to some of the COVID-19 research that's going on at Queen Mary. There's quite a bit, but I wanted to highlight just a couple that I think are important to note. The first is work that's being done by Professor Anya McKnight. And so she actually delivers some of the immunology lectures in the MSc Regenerative Medicine in the autumn. And so the work that she's a virologist by training, and she's recently published this paper in The Lancet that looked at the prevalence of COVID-19 in asymptomatic healthcare workers. And one of the kind of key takeaway messages from this was that the number or percentage of people with, or healthcare workers with COVID-19 that were asymptomatic mirrored that of the general population. And so it suggested that the healthcare workers were being adequately protected within the hospital, and that they were no more likely to transmit the disease to the patients. So this was an interesting piece of work, and they were able to set this up quite quickly, given their background in virology, but also the research facilities within the Blizzard that are set up for what we call level 3 biosafety. Another bit of research that's I think is particularly interesting is work being done by Professor Andre Kang in the Institute of Dentistry. So Andre is, or he has developed these antibodies that have a light-emitting enzyme attached to it, and these are called glow bodies, but they're very small, but they also produce a very large amount of light when triggered with a substrate. And so this technology was initially developed for multiple sclerosis research, but he's been more recently adapting that for COVID-19 diagnostics, and using this for very highly sensitive antibody detection. And then the last bit of research that I'd like to highlight is this new study that's being started, where they're looking at the risk factors of COVID-19. And this is probably one of the most comprehensive and the largest studies within the UK. And it's being led by Professor Adrian Martineau. And here they're aiming to recruit 12,000 patients and do a really in-depth analysis of what are the factors that put people at high risk and how can we potentially stratify them and provide the best treatment for those different groups. So again, this is a more public health aspect, but I think quite interesting and hugely beneficial to the healthcare sector. So that was just a really quick summary of some of the things that are going on at Queen Mary. Hopefully you find them interesting or exciting. I know I do. And just to summarize what I've talked about, we've looked at the development of next generation tissue models through these organ-on-a-chip and 3D bioprinting technologies, and some of the work, really multidisciplinary approaches to tackling COVID-19. And I think, together these really highlight the fact that research at Queen Mary is at the forefront of innovation and discovery. So with that, I'd be happy to take some questions, and hopefully if you are finding this interesting and exciting, we look forward to you joining us on the program next semester. So let me just come out of the shared screen and switch over to the Q&A. So if you do have any questions, feel free to pop them into the Q&A section. These can be either about the program itself or the research that's going on. Okay, I see one question from Anna. So she says, would you say it is still worth it for international students to move to London, even with only having online education in the first semester? Yeah, that's a really good question, and it's very difficult to answer. And I can't say for certain. I mean, I think there will certainly be advantages of moving to London, and you will have some interaction with other students and teachers, but at least for the first semester, it won't be the same as it normally would be. And I think that's probably a decision that you need to make yourself, and what you value, and is it worthwhile to you. I would say that for those students who can't come to London in the first semester, all of the education and all of the training can still be delivered and obtained remotely. So we're kind of putting in or setting it up so that even the students who aren't here can complete all of their training for the first semester remotely. So yeah, it's difficult for me to say because on one hand, we don't know how things are going to develop between now and September. And also it's a matter of, I guess, balance for you, whether, that level of interaction is worth the move. Okay, we've got another question for Farhan. Will the first semester be completely through online education? It won't be entirely through online education, but it can be if it has to be. So for example, if we have small group tutorials for those students who are on campus, those would be in person. And for those students that are not on campus, then that would be online. So it's going to be this kind of blended mix of online and in person. So Farhan has another question about whether this will impact the tuition fees for the course. It won't. The tuition fees that are published on the website will be the same regardless of your on-site or online. I think there's another question in the chat. So it says, what sort of research topics will be available for top master's students for the 3 months research near the end of the year? Also, are the fees going to remain the same with regards to the fact that the teaching is now more blended or a lot more online? So I think I answered the question about the fees. They will stay the same. Our hope is that we can move to a fully on-site delivery in the second semester, and then also have the research projects in the labs as they would normally be. Of course, that all depends on how things progress, so we can't guarantee anything at this point, but that is our hope. And then in terms of the research topics, so these cover a wide range of tissues, cell types, diseases, and approaches. And so we basically put out a call at the beginning of the year to any researcher at Queen Mary who is doing regenerative medicine related research and would like to supervise or host a master's student. So this can be something as fundamental as trying to differentiate pluripotent cells into a specific tissue type. So for example, Young Yao Lin, who is here in the Blizzard, he's interested in developing an in vitro model of muscular dystrophy using induced pluripotent stem cells. And so he's developing methods for genome engineering of these cells, and then also methods to differentiate them into muscle cells. So that's kind of one topic. Another would be more applied. So some of our colleagues in engineering are building biomaterials or hydrogels for culturing or repairing different types of tissues. So Elena Azevedo in engineering, she's particularly interested in using self-assembling peptides to repair tissues such as the cornea. So hopefully that gives you a little bit of a range of the breadth of topics that we cover. I see another question. How can we defer our offer of study at QML and until when? So if you do decide that you would like to defer your offer, you can do that, I believe, for up to 2 years. And you just need to contact the admissions office and they can set that up for you. One thing to bear in mind with the deferral is that if the tuition fees go up from 1 year to the next, then you would be expected to pay the tuition fees for the year that you enroll, not the year that you apply. So that's one maybe caveat to deferral, is that the fees may go up the following year. Okay, I see another question. Oh, sorry, I think that's 1 year for deferral. I think it's, so just, yeah, correction to my last answer, it's 1 year for deferral. Okay, we've got another question from Farhan, and says, once we join the university, can we join a lab and work under professors right from term 1? You can. It's not necessarily encouraged because the program is quite intensive. And so it's intended to be a full-time study for the top components. And so, you know, while you're perfectly free to contact professors and work in their labs, kind of on a voluntary basis, I wouldn't personally recommend it, particularly in term one while you're trying to kind of get settled in the way of studying and the format of the course. Ashwin says, can we shadow? So along the lines of my previous answer, you can. I would probably be cautious about how much shadowing you do, because the talk component is quite intensive, and it's probably best to focus on that, at least at first, and get settled before adding on any other additional activities. Yeah, Ashwin says clinicians while studying for the course. Yes, this applies both to research and clinical activities. So Muhammad asks if I can explain about the scholarship opportunities for international students. So there is a page on the Queen Mary website. I can put that in the discussion chat, but you can find it through how to fund my studies on either our course webpage or the university website, and that has a list of potential scholarships that you can search for and that you might be eligible for. One thing I would say with that is that it might be a little bit late in the year to apply for scholarships. Usually those have deadlines kind of in the year before or earlier in the same year. So it might be a little bit late for some of those, but do take a look on the Queen Mary website. There is a list of those there. And I'll see if I can put up a link for that at the end. And so one other question from Farhan, he says, what are the accreditation of this course? I'm not sure I know what you mean by accreditation. So it's not a clinical course, so there's not a formal accreditation in terms of clinical training. But I mean, it is a master's degree, and it's the UK does review all of its major universities, so it is a major UK university in that form of accreditation. But I think aside from that, I'm not really sure. Okay, so Mohammed has another question about the scholarships. No, there aren't particular scholarships for this program. The ones on the website are a more general Queen Mary wide or UK wide or international scholarships that you can apply for. Hopefully that answers your question. But if it's not clear, feel free to get in touch afterwards, and we can discuss that. And so one other question, when is the course date fixed? So let me just double check I get the dates right. So the course does start in September. So the first day of teaching this year will be the 21st of September, and so that is fixed. And then the week before, probably the week of the 14th, there'll be several kind of induction and registration activities. So I would advise you kind of plan to arrive in London or on campus if you are doing so the week before, so the week of the 14th, to kind of get settled and get everything set up for classes starting on the 21st. Okay, I don't see any other questions. Maybe I'll give you, one second or so to put any final questions in. And if not, then we will probably wrap things up. This session was recorded, so if there are things you want to go back and review, we can make this available. And as I mentioned before, there are, there's one other question. So last question, what are the quarantine measures for students who arrive in the UK? That's also a very good question. At the moment, the students arriving or people arriving in the UK from other European countries where there is a reciprocal travel arrangement, I think that starts on the 6th of July, won't need to quarantine. So like Germany, Italy, Spain, they won't need to quarantine, but that's what it is currently. And it's unclear what things will be once we get closer to the start date. So I'd encourage you to keep an eye on the UK government website. And also the Queen Mary website. And just want to note that they will get support from the Global Engagement Office. So they'll be able to advise you on, if there is a quarantine from the country that you're traveling, how long you may need to arrive beforehand. Okay, great. Well, thank you everyone for logging in today. Hopefully you found that informative and interesting. If you do have any questions, feel free to get in touch via email. We're happy to answer them. And like I said before, hopefully we'll see you here or either online in September. Okay, thank you and have a good day.